Hello, I'm Helen Windsor from Pharma IQ, and today I'm joined by Bill Skinner and Toby Winchester, Senior Principal Scientists from Pfizer. Thanks very much for joining us today. Now, I'd like to start by asking you about your roles. So, Bill, in data management, what departments, processes and people do you support in drug discovery materials management?
At Sandwich, we have a small team within materials and data management. Our team focuses on the data management, and we primarily support the materials management functions that exist in discovery at Sandwich. So these will be the order fulfillment systems in discovery, plate provision for high throughput screening, and the global liquid sample store at Sandwich. We also provide support to colleagues that input into those systems and receive samples from those systems. We share support globally with colleagues in the United States, and we also get involved in any sample logistics type projects that are taken on locally or globally.
Toby, can you give a little background on your role in running the compound management logistics operations at Pfizer?
Okay, my team work in one part of that sample logistics at Pfizer. Even at Sandwich, we have basically two screening areas, one HTS, which I'm not involved in, and one in the SAR generation, which I am involved in. Difference mainly being in the vast number of compounds that the HTS have to look at after, whereas we have to look after an awful lot of screens. So different problems, using similar equipment and usually the same compounds.
Looking at the big picture of compound management, years ago everyone wanted to bulk up, the bigger the better. Today it seems companies are doing just the opposite, making their files smaller, more compact. Please can you tell us your view on this trend, how it's evolving at Pfizer, and what this means to you in compound management, Toby, from a customer's point of view?
The research units at Sandwich are very time pressured. So screening a very large file, as in a standard HTS, is now not as popular as it was. So it's much more going towards subset screening and straight into literature leads to get the projects running as quickly as possible.
And Bill, what does this mean in data management? Can you explain how and why you're doing this?
Yeah, we've done a lot of work over the past few years. I mean, screening absolutely everything in high throughput screening, I think, is something of the past. The work that's been done with colleagues in computational chemistry has shown that if a series is going to be active in your screen, you don't actually have to test all the compounds you have in that series to get a hit. So it's finding the right balance between reducing the number of compounds and making sure you've got a really good chance to identify a hit if it's going to be one in your screen. So we've been writing software and algorithms to help us identify and reduce the number of compounds in our primary screening file.
We hear companies increasingly talking about implementing the automatic release of compounds for screening. Firstly, Toby, what is the reason that you've pushed this?
This is all down to timelines. We are under immense pressure to get projects running as quickly as possible and results from assays back to chemists as quickly as possible. In order to make this happen, we've implemented lots of changes, including very fast and reactive software systems to get the compounds to the screens and the right screen as quickly as possible.
And Bill, could you tell us a bit about this initiative, what it takes to set it up, what technology, software and people need to be in place, and what it means to overall store to screen process?
We've spent quite a few number of years putting together or integrating our internal systems. We've got a global ordering system and we've extended that so that people that actually do the screening can determine what they require in terms of they get into the lab. So the materials they receive are ready to go straight into screening. That then goes on for the project leaders to group together and identify what work or what screens they need to do within their project to identify their compounds and their strategy in how do they take leads forward. There's the submission angle from the chemists for new compounds and contract research organisations. Once these screens or panels of screens have been set up, they can then be instructed what to submit their compounds to, depending on the project. We then have this holding area which gives people within the company, both in Sandwich and in the States, the flexibility to decide how they want to use their resources. One of those options is to have everything automatically released on a schedule. Another one would be for the biologist to go in and say, I want to test so many compounds. Another would be for the chemist to go in and say, I need to push everything that we synthesize to go to a screen. And the final stage is actually integrating that with our sample management software to guide operators into what compounds to pull and when and what to do with them.
And can you offer a view on small molecules versus biologics?
Yes, this is another evolution of the research process. We have spent a lot of time working on small molecules. The future is probably both biologics and small molecules. We are compound management. There's no reason we shouldn't be able to, apart from the fact all our legacy systems only support compounds, small molecules.
Now, Toby, how small is small? Could you describe the evolution at Pfizer of going nano, i.e. historical volumes to today's volumes, and what that means to overall dispensing, screening and or drug discovery process?
The compounds plates that we produce in the SAR generation part of sample logistics have predominantly been large volumes, 35 microlitres of DMSO solutions is usually what we start with. We're now going to an area where we're talking 100 nanolitres is what we need to prepare samples with, and also what we are delivering to our customers. The main reason for one of the main reasons for this is pushing the hours that are needed within a screener's life to do further dilutions of the plates. If you give them a smaller volume, they have to do less dilutions. That saves them precious time, allowing them to do more science.
Toby, acoustic dispensing is having quite an impact on compound management. What are your recent learnings in this area, and what problems do you experience when bringing in this type of screen, i.e. temperature control, etc.?
So as you move to smaller and smaller deliverables, you encounter new problems. Fortunately, from a Pfizer point of view, we're quite late into the acoustic world. The beauty there is a lot of the lessons have already been learnt. But recently we've just taken delivery of a new system, and the fluctuations in the temperature of the laboratory are causing differences in the DMSO viscosity, which hence makes differences in the volumes that these systems are able to dispense. We're getting around that, but it was a problem we didn't foresee when we were first installing the system.
And I understand Lean Six Sigma is being applied in compound management. Could you elaborate on that? And do you see it as being a useful tool?
The evolution of the systems has not just been from a customer asking us to do something, but trying to predict what people need in the future. One of those prediction tools is definitely the Lean Six Sigma tools, and basically getting the customer what they want, even going as far as asking them actually what they want.
And again, Toby, from a logistics or lab perspective, what technologies do you see shaping the future of compound management in the next 2 to 3 years?
I think an awful lot will be based on the acoustic technology. That will be the major player, I believe. But there are gaps in those systems. They only deliver small volumes. there still needs to be a larger volume. So whether the acoustic systems evolve to be able to do larger volumes, or some other system can come along to work in a similar way. The other big gap that we see at Sandwich is you can QC your machines within an inch of their life, but you can't tell whether the compound has actually reached that final destination plate. If there was some technology out there that could detect whether your compound was actually in your final plate, then the assay has 100% reliability in the product they get from us.
Bill, from your point of view and experience, how can biologists and chemists work together to achieve better results?
I think we're already working close together in Sandwich. The research unit teams communicate very well, and our system that we've developed allows for the cultural difference between working between biologists and chemists at different sites. We've mentioned before the ability to push and pull, and where there's a service department like a pharmacokinetics, there's the ability to schedule work which allows those people to make better use of their time and plan better. I think it's a case down of planning the project and having a flexible system where all the requests going in, whether they're for new chemical entities or whether they're following up on previous work, get pulled together.
And Toby, what is your viewpoint on this?
Well, I work very closely with Bill, so it is that same area. We've worked closely on this for probably 10 years now, and so we have very similar viewpoints, I'm afraid.
And finally, working together to operate a smooth running compound management facility, what have you learned as a team, and what advice might you give to other compound management teams?
I believe it's important to have a team together that has a very broad knowledge, both in terms of the operating system. So obviously you need people who can operate the software, who can operate the technology. But on top of that, you need somebody with a very good understanding of the whole process and the data flow behind the process. This gives you the ability to respond rapidly to issues. A piece of equipment breaks down, it means that your data may not have been written. You need the ability to get that in place. And also, we're working in some areas on the front edge of technology. So when somebody comes along that wants to test a new plate format or the way they want their sample, it's important within that team to have access to somebody who can help make that happen without having to go through the whole life cycle process with a business technology colleague.
And Toby, to finish, do you have anything to add?
Yes, it's not just having a basics of the IT strategy and even the materials management strategy. You also need a customer focus. You also need the engineering backup for all the automation. You need a fully integrated team, not just a compound management team or not just an IT team.
Toby, Bill, thank you very much for taking the time to speak to me today.